Now, we are going to look at the biotransformation of DNT. Biotransformation of DNT. And DNT, the full meaning is 2,6-dinitro what? Toluene. 2,6-dinitro-toluene. 2,6-dinitro-toluene. Now, this 2,6-dinitro-toluene is a chemical substance that is hepatocarcinogenic to male rats. Now, we are going to look at how it is biotransformed. First, if the 2,6-dinitro-toluene, look at the structure. If you are naming this 1, 2, 3, 4, 5, 6, 2,6-dinitro, and this is toluene. So 2,6-dinitro-toluene. First, 2,6-dinitro-toluene can be converted and can be hydroxylated to 2,6-dinitro-benzyl alcohol. And who can tell me the name of the enzyme that can catalyze this reaction? No. From here, we are just adding hydroxy group. Cytochrome P450. So cytochrome P450. Good. Now, so the first step in the liver cell, remember all these xenobiotics are transported to the liver, right? Now in the liver, cytochrome P450 will first of all hydroxylate this, add hydroxy group to this, and we have, is no longer toluene, but what we are now having is benzyl alcohol. So we'll be having 2,6-dinitro-benzyl alcohol. Now, after that, this 2,6-dinitro-benzyl alcohol can have two fates. It can be converted by alcohol aldehyde dehydrogenase. So what's the name of this? 2,6-dinitro-benzoate, right? So this is 2,6-dinitro-benzoate. And this 2,6-dinitro-benzoate is water soluble. So as it is water soluble, it can easily be excreted through the urine. But if it follows the other pathway, that is where the problem comes. Remember, the first thing we said, the 2,6-dinitro-toluene first will be hydroxylated to what? 2,6-dinitro-benzyl alcohol, right? Now the 2,6-dinitro-benzyl alcohol can be converted, can undergo oxidation to 2,6-dinitro-benzoate. Remember, alcohol, first of all, if it undergoes the first oxidation, to form aldehyde, right? Benzaldehyde. If it's further oxidized, to form what? Carboxylic acid. So that's that. Now the next pathway you can follow is conjugation reaction. Remember, this step here is this phase 1 metabolism or phase 2 metabolism. Phase 1, right? Because phase 1, we're looking at hydroxylation, oxidation, hydration, reduction, right? We are not combining it, so it can follow just the phase 1 reaction and it will be excreted, or it can follow the phase 2 reaction also. So after it undergoes hydroxylation, remember, the purpose of phase 1 reaction is either to add the functional group or to expose an already existing functional group, right? Also, the purpose of phase 1 is either to make it more soluble so that it can be excreted, or to prepare it for phase 2 reaction. So in this case, after forming this can follow another phase 1 reaction and it's excreted, or it is prepared for a phase 2 reaction. So the 2,6-dinitro-benzyl alcohol now can undergo conjugation with glucuronide. And remember, in conjugation with glucuronide, what is the donor? UDP what? Glucuronate, right? UDP glucuronate. And the enzyme is always what? UDP glucuronide transferase or glucuronosine transferase, right? Alright, so here we use glucuronide transferase. So glucuronide transferase, we transfer glucuronate from UDP glucuronate, right? So you can indicate it that UDP glucuronate. Let me use that. And what will be going up is UDP, right? So the glucuronate now attaches to this. And remember, when it's attached, you no longer call it glucuronate, but you call it what? Glucuronide. So we now have 2,6-dinitro-benzyl alcohol glucuronide. Just the name of this, then you add glucuronide as a conjugate. Now this stuff here can be transported through the bile to the intestine. So it is transported from the liver now to the intestine. And remember, yes, in our last class we said that in humans, most of the microflora we have is located in the large intestine, right? Now it can be transported to the large intestine, and in the intestine where we have the majority of the microflora there, microorganisms can act on it. Remember, we're looking at xenobiotics whose toxicity is dependent on microflora metabolism. Now, this same compound here, this 2,6-dinitrobenzyl alcohol glucuronide, is what is transported to the intestine now. If you look at the structure, they're the same thing. Now, this 2,6-dinitrobenzyl alcohol glucuronide will be acted upon by microflora reductase. It will reduce one of this nitro group to an amino group. If you look at it, the only thing that is changing from this structure is this is reduced to this. Remember, reduction is addition of hydrogen, removal of oxygen. So this is reduced to this not by our enzyme now, not by the human enzyme, but the microflora enzyme. That's what is doing that. Now, after that, the next thing is still microflora beta glucuronidase. Beta glucuronidase is an enzyme that removes what? Glucuronide. So the microflora beta glucuronidase will remove this glucuronide, this conjugate, and we'll be having our alcohol back. Do you get that? Yes. Then the next thing, this 6-amino-2-nitrobenzyl alcohol can have two fates. It can be converted to 2,6-diaminobenzyl alcohol, or it can undergo reabsorption again from the intestine. Now, reabsorption, remember, in the large intestine there, from the large intestine, the last stage of digestion is in the small intestine, right? Undigested will move to the large intestine, and from the large intestine, it can be stored at the rectum and excreted. This can be excreted because this is the large intestine. Or instead of that, it can also be reabsorbed back into the intestine. Can be reabsorbed back instead of being excreted, can be reabsorbed back. And if it's reabsorbed back, if you look at this structure, you see it's the same thing as this, right? It's the same thing as this. Now this can undergo further metabolism by cytochrome P450, trying to break it down. Now cytochrome P450, the main function of cytochrome P450 is hydroxylation, addition of OH. So cytochrome P450 can hydroxylate this, adding OH here. Do you get that? So 2N-hydroxyamino, said 2N-hydroxyamino, because if you are counting, this is, if you look at this 1, 2, 2N-hydroxyamino, 6-nitrobenzyl alcohol. Now this also can undergo, what type of reaction is this? Let me see who's thinking first. So this can follow two paths. It can undergo acetylation or what? Or sulfation. So it can undergo acetylation or sulfation. So if this undergoes acetylation, we have, now it will not be N-hydroxy again, but what? N-acetyl, right? Is it not N-acetyl? Or it undergoes sulfation. So there we'll be having what? N-sulfo as a name. Then these two can bind covalently together. Before I go further, let me take that again. If you look up, what we are having is 2,6-dinitro what? 2,6-dinitrotoluene. Now this 2,6-dinitrotoluene here, we said first cytochrome P450 can act on it, and we said the main function of cytochrome P450 is what? Hydroxylation, addition of hydroxy group. So if hydroxy group is added here, the name of the compound is no longer 2,6-dinitrotoluene, but what? 2,6-dinitrobenzyl alcohol. And the 2,6-dinitrobenzyl alcohol can have two fates. First, it can be oxidized, further oxidized to 2,6-dinitrobenzoate, or it will undergo second phase reaction, which is conjugation reaction, right? Now, the 2,6-dinitrobenzyl alcohol can undergo conjugation with glucuronate. And if it undergoes conjugation with glucuronate, we know that the donor in glucuronidation reaction is what? UDP glucuronate, and the enzyme is what? UDP glucuronose transferase, right? So if that happens here, glucuronate will be attached here, and if glucuronate is attached, the name is now what? Glucuronide. So glucuronide is attached here, and we have 2,6-dinitrobenzyl alcohol glucuronide. Now from here, we said this substance, this compound here, is transported through the bile to the intestine, and specifically the large intestine, because that is where majority of the microflora is in human beings. Now as it gets here, this compound gets here, the microorganism will start to act on it. And as it acts on this, the microflora nitro reductase, that's the first enzyme, will reduce this nitro group to an amino group. After that, microflora beta glucuronidase will remove this glucuronide we added. So it will remove this glucuronide, and we have 6-amino 2-nitrobenzyl alcohol. This can have two fates. It can either be further reduced. This other nitro group can be reduced to this, or it is reabsorbed. And if it is reabsorbed, cytochrome P450 can further act on this. And the main function of cytochrome P450 is what? Hydroxylation. So now we already added OH here. We are not hydroxylating this place again. So we hydroxylate this to get this. Now this can have two fates. It can undergo sulfation, another phase 2 reaction. If you look at what is happening here, it's just like this is a new xenobiotic that is undergoing first phase 1 reaction and then phase 2 reaction. So it can undergo sulfation to form this, or it can undergo what? Acetylation to form this. Then the two of them can covalently bind together. That's that. I want you to understand this pathway, and there's something I want to say about that. But before then, any question? If you don't ask, I'll ask you that particular question. Yes.